concluded the previous video with an introduction to the concept of antigen-antibody interaction. Let's have a look at this concept in detail in this video. Any idea how this interaction takes place? The answer lies in the structure of the particular antibody. Antibodies, also known as immunoglobulins, are denoted with the abbreviation Ig. They are Y-shaped protein molecules which are made up of different peptide chains. Here we have a complete representative structure of a typical antibody. This structure of antibody is basically studied by dividing it into two parts: the constant region and the variable region formed by these peptide chains. As the name suggests, the constant region means the peptide chains remain the same in all antibodies. On the other hand, the variable regions will have peptide chains that can get altered as per the requirement. And what will this requirement be? That's right. The requirement will be the shape of this site depending on the structure of the antigen. Needless to say, the antigen binding site will be present in this variable region. There are many detailed parts and structures present within these variable and constant regions, but we will keep them reserved for the higher grades. For now, all we need to know is that all these parts together constitute a complete antibody. In us, humans to be precise, we have different types of antibodies. These include IgM, IgG, IgA, IgD, and IgE. For now, let's just know their names. And let's understand how any type of antibody interacts with any antigen that is designed to destroy. Antigens, as we know, are the molecules which are capable of triggering an immune response upon entering the body. So when an antigen enters the body, the immune cells secrete antibodies specific to the antigen. That means, if this is the antigen, then this antibody with the binding site matching its structure will be produced. And once the antibody encounters this specific antigen, it will quickly bind to it somewhat like this. This is the antigen-antibody interaction that we've been talking about so far. And such interactions give rise to a cascade of reactions which ultimately trigger the immune response. So imagine this is a pathogen with these antigens at its surface. On encountering this one, the immune cells will make antibodies specific to these antigens, and thus an interaction somewhat like this will take place. And as a result, the immune system will get further activated. This step may be extremely critical when it comes to the fighting of the infections in the body. But how do we understand it practically? There are several wet tests performed in the laboratories that give us a glimpse of such beautiful reactions. Elisa test, for example, is one such beautiful wet test which is based on the principle of antigen-antibody interactions. Let's see what this test is all about. ELISA, which stands for Enzyme-Linked Immunosorbent Assay, is a test in biochemistry that helps in the assay of a liquid sample to qualitatively and quantitatively measure the particular component present in it. The principle is quite simple. It's just the interaction of antigens with antibodies. ELISA is usually carried out in a 96 well plate which looks somewhat like this. The bottom of this plate will be coated with a known antigen first. Then, a sample containing the antibodies is added to this. But there is a small twist here. The antibodies are linked with specific enzymes. These specific enzyme-linked antibodies will bind to the antigen. And what will happen then? On adding the substrate at the end, the enzymes linked to the antibodies will bind to it and thus show a change in color. The intensity of color change helps detect the amount of the analyte we are looking for. This is just one type of ELISA which is known as direct ELISA. But we will restrict ourselves to this much as we will now deviate towards the concept of immunity. Now that we've glanced over a few more useful concepts related to immunity, Let's understand its types in general. Immunity in every human is divided mainly into two broad categories. One is called the innate immunity, and the second is called acquired immunity. Innate, as the name suggests, is a non-specific immunity present in an individual right since the time of birth. On the other hand, acquired immunity is the one which is gained over a period of time or which is developed during an entire lifetime, as and when we encounter the pathogenic invasions. This type is highly specific in nature. Let's get into the details of each type in the upcoming videos. Till then, do subscribe to our channel for more cool videos on such interesting topics.